Hello everyone, today I'll be talking about rheological characterization of covalent adaptable thioester networks. The objective of this work is to characterize covalent adaptable thioester networks for their applications as cell delivery vehicles in tissue engineering problems. Covalent adaptable networks have come across as promising candidates for tissue engineering as they can mimic certain aspects of extracellular matrix. As in, they can rearrange themselves under external stimulus like shear, light, or pH. Cells residing inside extracellular matrix are constantly applying forces on the matrix to rearrange it and to perform cellular functions like spreading or differentiation. Covalent adaptable network, when used for cell encapsulation, can also allow cells to perform all these functions. In order to tune their properties for tissue engineering problems and enhance cell delivery, it is very important to characterize macrostructural and microstructural properties of these materials. We use bulk rheology to measure equilibrium swelling and to measure stress relaxation properties of the material. We use MPT to understand how the microstructural changes take place in the material over time. Now we form thioester network by photopolymerizing 8-arm PEG thiol and PEG thioester norbornene in presence of photocatalyst lab. When there is excess thiol present in the network at alkaline pH, the thiol can form thiolate ion and attack the neighboring thioester bond, which leads to the formation of new thioester and thiol, resulting in the rearrangement of the network. We first use bulk rheology to characterize a material where the usual principle of bulk rheology is to apply deformation to the material and measure response. It could be either over time, or we could apply deformation at different rate and then measure the properties like storage modulus. We first measured experimental swelling in the material by frequency sweep measurements. Uh, and we measure storage modulus for samples which are incubated at pH 8.4 for different length of time. The data shows that the average storage modulus for samples which were incubated over 1 hour is within each other's error. Now we measure stress relaxation properties of the material. Stress relaxation usually shows how the material dissipates the applied stress by rearranging itself and how quickly it can do that. When there is no excess thiol present in the network, there is no stress relaxation seen in the material. But as we increase the amount of excess thiol in the network, the stress relaxation rate also increases. Now we measure microstructural properties using MPT. MPT usually measures Brownian motion of the fluorescently labeled probe particles which are embedded in the sample. It captures the Brownian motion using the video microscopy, and then we use a particle tracking algorithm to get the positions, and from there we use probability density functions accounting for Brownian motion to get the trajectories of these particles, and from there we calculate ensemble average mean square displacement of these particles. The graph of the logarithmic slope of graph of MSD versus lag time alpha is important in telling the phase of the material. When alpha is equal to 1, the probe can move freely through the network. And when alpha is equal to 0, that means probe is completely trapped inside a sample spanning network, meaning it's a gel. Between 0 and 1, it could be viscoelastic sol or a gel. Now, our MPT experiments were performed at physiological pH, and we can see there is fluctuation in the alpha value for the samples over the time, which these fluctuation could be resulting from the bond exchange reactions. Now, to conclude, we were able to use MPT and bulk rheology to measure the properties of these networks. Frequency sweep experiments show that the material obtains reaches equilibrium swelling modulus within 1 hour, where stress relaxation experiments show that these materials have tunable stress relaxation properties. And as we increase the excess thiol in the network, the stress relaxation rate increases. The MPT experiments show that these networks rearrange themselves at physiological pH, which result in the change of mobility of the probes over time, and which also results in the fluctuations of alpha value. Thank you.